Now, I'd like to kick off the webinar by introducing our speakers. Manoj Dadlani is the Chief Executive Officer of CosmosID. He's a Cornell University graduate in biological engineering and previously served as the head of the U.S. branch of Applied Value Management Consulting. Since then, he's transitioned to the investment side of the Applied Value Group, which is the lead investor for CosmosID. Dr. Rita Colwell is the president of CosmosID and serves as a distinguished professor at both the University of Maryland at College Park and at Johns Hopkins University Bloomberg School of Public Health. Dr. Colwell has a very impressive and extensive background, having authored and co-authored 19 books and more than 800 scientific publications. Her bio goes on, and you can read more in the box on the left of your screen. With over 40 years of research, innovation, and scientific leadership, Dr. Colwell has a keen understanding of the ecology of infectious disease and the use of advanced technologies to halt their spread. And with that, I'll turn the presentation over to Dr. Rita Colwell.
Thank you very much. It's a great pleasure to speak to the audience today. I wish you all a good day. We'll be discussing the applications of metagenomics for public health, food and water safety, and epidemiology. In other words, an expansion of the capability of metagenomics. The usual disclaimer, what I say is my opinion and not the opinion of Illumina, and any of the uses that we discuss have not been approved by the FDA, so I just want to make that general disclaimer. What we're going to be discussing is the comprehensive microbiome data analysis, the platform that's available. We analyze microbial data sequences. We have a huge proprietary database, over 150,000 bacteria, viruses, fungus, protists, including parasites, and it's very rapid. We can run analyses in minutes, and the informatics is really the best in class, as I'll explain later in the lecture. We identify to subspecies, to strain level, and we can do an accurate abundance measurement and characterize specific genetic traits such as antibiotic resistance and virulence factors. The platform is agnostic, short or long reads, and we make sure that the version that you use is a standard version, and also we maintain the software and the databases. We keep them up to date and make sure that they're timely and accurate. It's really a powerful bioinformatics capability with curated genome databases, and with this comprehensive set of data, which we organize very specifically into phylogenetic trees. Having learned a long time ago that taxonomy is kind of quirky, depends on whoever does the naming, and it doesn't really represent the organisms so specifically. So we've chosen to organize our data as phylogenetic trees. And we use a couple of types of biomarkers, those that are unique to the organism, and then those that are shared across the phylogenetic lineage. And we can customize the databases to specific applications as well. So I think that it's critical to point out that we can identify bacteria, viruses, fungus, protists, and provide specialized information as well, and characterize virulence factors, plasmids, phages, antibiotic resistance. So I think the other important point I'd like to make is that having worked in systematics for about 40 years, having written a computer program using phonetic data about 40 years ago, the important point for me is to make this tool available and easy for any user to apply. And so, you know, bioinformatics can be rather daunting. It may, in the minds of some, require, you know, specialized computer guys, but really, the way we've organized this, it's simple to use, but still highly sophisticated. And it's do-it-yourself bioinformatics, where you can upload the data, you get your analysis in minutes, and we have terrific visualization tools that allow you to see the analysis. Now, I want to make sure that the listeners understand that we've done a lot of applications, so this is not something novel. We've been working on this for 10 years. We have a very important contract with the Food and Drug Administration where they use our platform in the food safety aspect of the FDA regulations. And we also have a very nice collaboration with NIST where we have a metagenomics challenge that we have with NIST. We work closely with Illumina and with BaseBase. You can import your data and do analyses, which I think is very, very important. Only other point I'd make is that we are part of the Vice President Joe Biden, the former Vice President's moonshot family, so we're in that as well. Now, what we do is offer an end-to-end solution. Really, our forte is bioinformatics. That's where we excel, and we can provide tremendous, I think, advantage for investigators. But because it's important to be able to provide for individuals who want to do analyses, we can do the analyses starting with helping you set up the study. We can provide information and advice on how you collect the samples, how you prepare them, and then, of course, we can do sequencing as well, and then provide the analyses and the informatics. This can be as personalized as you need, or it can be simply shoot the data into the cloud and get the analyses as you need. And so we do provide the cloud application, and that is because we collaborate with small pharma, individual investigators who are doing bioinformatics and metagenomics. So with a cloud app, this allows you to load up your data, FASTA or FASTQ, and get the analysis done and download with the interpretations provided. That is, we don't just shoot the data out, but you can get it in a variety of formats, which I'll mention as we move on in the talk this morning. Now, I think it's important to point out as well that we have analyzed tens of thousands of samples, food, studies of oil pollution in the environment, cosmetics, a subway study, etc. But what we have learned is that for us, being able to apply machine learning, it allows us to tailor the output. And I think that is rather important to emphasize. So whatever it is that you want done, we can tailor the sample system and the output to your needs. I think it's important to point out as well that we have gone against all of the available bioinformatic tools, and using the data in a paper that's cited in the lower left-hand corner of your slide, using those data, we did analysis and came out really best of class. We can identify down to species and the subspecies level, and we have the highest accuracy and the best precision. These are factors that are very, very critical. The statistic is the F1 harmonized mean of precision and recall. So it really provides you with accuracy and specificity, and I think is important to emphasize compared to all of the other available tools that are open tools as well as any other proprietary tools. So I'd like to emphasize that it's a cross-disciplinary microbiome platform. For each of the verticals here, that is diagnostics, exploratory work by investigators and groups, public health screening, etc., you see listed along with each relative publications, but the tools are in fact the ones that I'm describing today. This allows us to tailor the database for each of the specific needs, and you can apply to each of the publications for details in the specific applications. Now, the technology has been featured in a study of leading companies in microbiome analysis, and I can't help bragging a little bit by pointing out that under the genomics category, where we can deal with just about all of the applications, we're listed as one of the top best of the startups. But the other point I would emphasize is that Science magazine featured CosmosID with respect to cross-disciplinary microbiome discoveries. I would say particularly the food safety application was noted. So that's just to point out that there's a lot of experience here. Now, I'd like to emphasize that the workflow is pretty straightforward. The sample is input, and we do work with this new technology, the Nextera Flex that Illumina has. It's a new product, and the technology is helpful. But we're also agnostic to the sequencing platform. You can select any one of the platforms depending on the need of the project and the outcome with respect to sequencing depth. But everything then can go into base space, can be imported. And once in the base space, you can streamline it to directly to the CosmosID analysis. And the kind of analyses we can provide is very expansive. Everything from simple tabular output to Krona to various kinds of listings and analyses. The metagenomics platform, if you note on the left-hand side, you can directly import the data from your base space account to do comparative analyses, and then you can also upload directly from a local machine, and that allows us to compare hundreds of samples and to do comparative analyses. I think that is just a point that I'd like to make, that it's a very highly capable platform and tool. I think one of the best examples is that a water analysis, which I'll give more detail in a moment, but this is a demonstration, a very quick demonstration of sewage water going into a water treatment plant for reuse. And we can do a multi-kingdom microbiome analysis. That means all of the bacteria down to species and strain, viruses, RNA, as well as bacteriophages. And then also we can do analyses of the fungus that are present, the fungi, and protists, particularly parasites. We have all of the pathogenic human parasites in our database. So this means that in analyzing a water sample, we can provide all the microorganisms that are present, from the bacteria, viruses, fungus, and the protists. And in addition, we can also provide the resistome, that is, all the antibiotic resistance coding genes that are present. This is done simultaneously. You don't have to do iteratively. This is what the output provides. And the virulence genes as well, virulome, I think we can call it that. So this provides a very comprehensive analysis, and I'll give a specific example in a moment. But again, let me point out that the output is user-friendly. You can get a stacked bar chart, you can get a heat map, as well as a Krona or principal component analysis, PCA. So you get a variety of outputs, and this will be demonstrated later in the session today by Manoj Dadlani. But I think it's important to point out that you get a very, very comprehensive analysis for the output. Let me give you some examples. First of all, let's start with a couple from public health general perspective. And I think one of the ones that is very interesting is a study we did collaboratively with the Singapore group of the biofilm environmental life science and engineering team there. We did a study of blowflies. What we found out really is that, no surprise, flies have dirty little feet. So in the next slide, I'll point out that analyzing the microbiome of the dissected abdomens, the head of the flies, the legs and the wings combined, and the thorax, we find that really the highest numbers of microorganisms, the diversity, the highest diversity, as well as numbers, was really associated with the legs and wings. So it is a fact that having flies land and walk all over something is not a good idea. We found out, too, that one of the very dramatic findings is that Helicobacter pylori is dominant, particularly in the legs and wings, but throughout the blowfly. And that's very interesting because then when we went on to do the next analysis, we found that not only is the Helicobacter pylori there, but the specific genes, the CAG11 genes, those that are pathogenicity islands, which provide enhanced virulence, the Helicobacter pylori, demonstrates not only can we show the organisms that are present, but also their capability in terms of virulence and antibiotic resistance. So again, not only do we identify, but we provide characterization of the microorganisms that are present. Let me give you an example now of water safety, some recent studies that we've done. First that I'll show is a study of a wastewater treatment plant done with colleagues in Saudi Arabia. This next slide gives the publication that you can refer to. This was a study where they were doing analysis of the wastewater to be reused. In Saudi Arabia, they have to reuse the water for agricultural purposes. They are not allowed to use it for direct drinking water, but they can use it for agricultural purposes. So the question was, how safe was the water that was going to be applied to crops that would be grown, particularly the hydroponic agriculture in Saudi Arabia. So the concerns really are what kind of microorganisms are present, pathogens, bacteria, viruses, etc. So in the analysis that was done, we were able to show that, if you show the next slide, that the wastewater treatment plant, after treatment, you can see on the left-hand side, this published study shows that we are able to determine RNA viruses as well as the DNA viruses. And this was very, very helpful because it could allow us then to determine the presence of human virus pathogens as well as plant and animal pathogens. What we found was that after treatment, in the treated effluent, the only viruses that were really abundant were plant viruses. So that allows us then to be able to inform the plant operators about the safety of the water. It's interesting because we're able to pick up the plant viruses that applying it for crops. If, for example, if you're growing peppers and then you find that you've got a lot of pepper virus still in the treated water, it's not a great idea to use it on that particular crop. So I just make that point to emphasize the fact that it's important to know the complete composition. The RNA sequences provides a complete viral composition, and this then allows us to better inform as to the applications that can be provided. Now, we've done another study which we're doing in great detail, and this is advanced treated wastewater biofilm study. This is done with Orange County Water Management Organization, the Water District of Orange County in California. As you well know, the drought in California has required that this work, which we've been doing with Manu Leddy, the first author in the publication, who has been extraordinarily great collaborator. What we're doing here is with the wastewater that comes from the sewage plant, it goes into the drinking water plant, and the Q1 is what we call the water that comes in from the secondary effluent. That's the coming in from the sewage treatment plant. So that, as it goes, the sludge goes out to sea, and the water goes to the advanced treatment wastewater plant that ultimately ends up with a product for drinking water. So what comes in is crudely filtered, treated with sodium hypochlorite, and then a microfiltration is done of the water, and then a reverse osmosis, and then it's irradiated, and then they do additional treatment before it ends up as drinking water in Orange County, Los Angeles area. So I think that last step is that they put it into they put it into an open water and groundwater area, so it filters down through, and they draw off the groundwater. Now that final step is very expensive and time-consuming, and the question is whether, after all the treatment from the original filter screening to the UV irradiation, is that safe enough to use as drinking water? So the answer we were able to show as follows. We looked at the Q1 influent water, and you can see the microbiome shows that just about everything, including the kitchen sink, is in that influent water. But after it goes through the millipore filtration, what's left are really maybe occasional opportunistic pathogen, but generally speaking, these are the water bacteria and soil bacteria that you would find in any tap water analysis that you pick up. So this allows us to be able to determine the safety of the water. In addition, what's very interesting is that there's a concern that the sewage water coming in would be adding antibiotic resistance genes to the water bacteria that you would find in tap water and in the environmental water that's being sprayed out and filtering through the wetlands to the groundwater. It turns out that, in fact, the antibiotic resistance genes are removed, and that you see from Q1, the bar chart I think is the most dramatic depiction. Q1 water has a lot of antibiotic resistant genes, but as it goes through each step of the treatment, from microfiltration to reverse osmosis, the antibiotic resistance genes are removed. And I think this is really critical because it then says that the output is not adding antibiotic resistance to the environmental water system, and particularly to the bacteria that are being released to the environmental system after this particular treatment. Now, this has been published, and we're doing additional work In fact, we received a National Science Foundation grant with Manoj and the Orange County Water District to continue the studies and do greater analysis of the RNA and DNA components to ensure that we have a complete capability, which we know we have, but to demonstrate the capability for analyzing both animal, plant, and human viruses present or being able to be removed by treatment in this water system treatment plant. Now, we also are doing a lot of work on food safety, and I think this is a really important aspect because we've been working very extensively over the last couple of years with the FDA. We've published a couple of papers, and we've got work in progress that we're now getting ready to publish with the Food and Drug Administration. They're the senior authors, and we're working closely with them. We've done an analysis of the Illumina platform, the NovaSeq, and this provides us with a very large data set. And we're in the process of developing an in-house laboratory rapid turnaround system for pathogen detection, food safety, and microbiome analysis. This is being done with the FDA. Now, one of the studies that we've done, which I think is very interesting, was the Bluebell ice cream event or problem that occurred where some individuals actually died from the contaminated ice cream. It was Listeria monocytogenes, and what we were able to show, and we published this with FDA, is a really nice study showing that very early, without any enrichment, in using a naturally contaminated ice cream test sample that was linked actually using the strain that was part of the outbreak. And this work was done with Andrea Otteson at the FDA. Andrea has been terrific in collaboration. And we were able to show that in this test system that without any enrichment or enhancement, we can pick up the actual agent. And we noted as the time of incubation that indeed there is, after 48 hours, we can pick up the organism. But the point that I would make is that what happens early in the sampling, we find that the Listeria is actually suppressed by the growth of Anoxibacillus flavothermus. So this flavothermus species inhibits growth, but eventually, with greater time of incubation, we pick up the Listeria monocytogenes. But the important point is that we can pick it up right away. We don't have to wait for incubation of 24 or 48 hours. We can look at the original sample and find the pathogen, and then be able to, with incubation, we are able to get a full analysis of the pathogen that allows us then to go the next step to for reconstructing the genome of the Listeria monocytogenes. We can then take the next step, which is to do a phylogeny and a tracking of the pathogen. That is, not only do we detect it very early, but we can actually reconstruct and assemble the genome, and that allows us then to do a really very extensive and comprehensive tracking of the pathogen in an outbreak and be able to determine the spread. So we've done a lot of work on antibiotic resistance, which becomes a problem because if animals carry bacteria in their gut that are harmful, cows or chickens, and antibiotic resistance, this is really important for us to be able to determine the source of the antibiotic resistance. And I show here the different types of examples that we can track the antibiotic resistance genes. We've worked with a variety of food matrices because we are building baseline data for different types of food, seafood, beef, crops in the field, specific melon, cucumbers, peppers, whatever, to be able to have a kind of baseline comparison of what would be, let's say, the expected flora, the expected microbiome of a radish, let us say, that will allow us then to determine what the anomaly may be with respect to a given sample associated with a food outbreak. And so we've sequenced about 1,000 samples to create this database. Let me now move quickly on to an example of some epidemiological studies. These have been fascinating and I think can be very instructive. Firstly, let me just point out hospital MRSA strain subtyping. If you notice up at the top, the P and H, I hope it's large enough, P means a patient isolate, and H means a hospital isolate. We were able, in this very rapid strain subtyping for molecular epidemiology, to detect clonal transmission utilizing the CosmosID tools. We were able to do rapid typing and run it through the pipeline and build a phylotyping that allows us to rapidly determine whether it's a hospital isolate or a patient isolate. And this allows us also to integrate metadata, that is resistance genes. Let me just point out that we were able to detect the same plasmid in a couple of patient strains, and I think different patients. So that, I think, is an important finding. And we're able to determine presence or absence. The green means specific genes are present, and white means that they were absent in that particular patient or that hospital isolate. So that gives us really a very nice capability. And then with this SNP tree, using the USA 300 as a reference clone, we were able to develop a very extensive phylogenetic tree that provides clonal relatedness. And this is a very elegant application that gives a lot of, I think, very, very useful, detailed kind of information. One of the important points to make again is that source tracking is critical. In this particular example, in collaboration with Amy Mathers at the University of Virginia and Chris Grimm, who's at the FDA in the Food Safety Unit, we were able to determine about 15 Klebsiella pneumoniae with a carbapenemase resistance, along with Aeromonas and Enterobacteriaceae clinical isolates. We collected this set, and we paired these with 32 sink trap isolates from biofilm in the sink trap. The point here is that they were trying to determine where this antibiotic resistance was coming from. They tried the usual swabs of equipment, walls, and so forth, and then got the idea that, this probably was coming from the sinks. And dismantling the sink and going into the sink trap, we picked up isolates. And then doing the comparison, the red are the patient isolates, the sink traps are blue, and we can actually track the isolates to the specific rooms and the specific sink trap. This provided a really very, very elucidating study, showing transmission from sinks in the patient rooms to the patients from the aerosols that it formed. Furthermore, we were able to demonstrate that the resistance was plasmid-mediated and plasmid-associated, and that provided us in with an analysis of the biofilms in much great detail. So I think this can demonstrate the power of what we're doing with respect to applications in food, water, and epidemiological analyses. We're doing in this other studies, but I think this provides a pretty good coverage, at least for you now to see an example of a demonstration. I'll turn this over to Manoj Dadlani, who will walk you through an example of how you do an analysis. I've given you a lot of information of analyses that we have done, demonstrated the fact that it's very, very useful. But now, if you want to use it, how do you do it? So Manoj, take it over.
Thank you, Dr. Colwell. So I'm going to go in and do a demo in a couple of minutes, and just a little bit of background. Here we're going to be showing version 2.0 of CosmosID. It will be available next week. It's in final stages of development, but you'll be able to, we're going to use that for the demo, so you'll be able to see the full functionality. What we've done in this new version is we've added in the ability to analyze 16S data, ITS data, and we've expanded our comparative analysis capabilities. And I'll show you that in a minute. And ultimately, you'll see the very fast, accurate, and secure platform that we've expanded on. From just a little more background, we'll go into the comprehensiveness of the analysis, and Dr. Colwell gave a lot of examples of output from the software. A little bit about security and version control. We have very strict version control. The results are very reproducible. We have high levels of top security encryption. We also have an appliance version if that's something that's required, although we've seen most people moving to the cloud. Recently, we've just set up a partnership with DNA Nexus to offer HIPAA compliance, CLIA compliance, general lab practice requirements, as well as ISO compliance. So we're very happy about that relationship with DNA Nexus and the ability to offer all different types of compliance that you can need. Finally, from a flexibility standpoint, we've also launched a command line API, which I'll show you a screenshot of in a second. And we have various different integrations with Illumina Basebase and other sequencing platforms to be able to integrate your workflow into any possible end-to-end solution. And we have the ability to do custom data. Here is what traditional bioinformatics has looked like. So this is our command line interface. It's Python-based. And this is generally when people talk about bioinformatics, this is what they think about. So we have this option, this ability to mine our databases and algorithms through the command line. But what I'm going to show you now is our do-it-yourself bioinformatics platform. So it might take a minute to buffer. Just a heads up that my internet connection is a little slow. I'm in a hotel room in San Francisco, so apologize for any lag there. And I'll just give it a minute to load. So I'll start off by just showing you we've updated our documentation. So if we click here, this is the new interface. If you click on the documentation, you'll see a very in-depth documentation, which will help you get started. Very easy to use, reference. And answer any questions that you might have. You can, of course, can reach out to us by chat or email, and we will respond very quickly to any questions to help you get going. So if we start off, I'll start off by showing you how to upload data. So two options. You hit the hamburger, you'll see you can either do an Illumina Basebase import, or you can upload data through our upload section of the app. If you go to the upload section, you can choose whether your file is Shotgun Metagenomics, Amplicon 16S, or Amplicon ITS. And then this box is a box where you can drop a file. So I'm just going to drop a file here, and it'll upload. You can put multiple files in there or just one. If you click on the box, you can look for files just like you do an email attachment. Alternatively, if you have your data in Illumina BaseSpace, you can log in, and there's a login button here, and you can connect your BaseSpace account and dig in and find some files. So once you have a file, you highlight it and you click import, and it'll import the file from BaseSpace into your account, and then it'll proceed to analyzing it. So there's a pop-up that'll come up saying that your file is being imported, and it'll move over to the dataset section of the interface. So this is the file importing, and the other file that is uploading is still uploading. So again, my internet connection is pretty slow, so we can come back to these files towards the end of the demo. Now, once you've uploaded your files, you can go in, and there's different types of file management that we've set up. So we have a folder system. So if you want to set up your files into folders, you can put them into folders. And to add a new folder, you would just click here and add a folder. Say new, you create it, and that folder would be set up. And if you want to move a data set into a folder, you would just highlight it, and you would move to, and you would pick the folder you want to move it to. So we can move that into the new folder. So now that sample has moved into this folder. We've also added a lot of features around metadata. So now you can add metadata to your samples. So if you just highlight it and click the metadata sheet, you can go in and fill in various types of metadata. You can also add custom metadata to your samples as well, which will allow you to track and manage your samples as well as do better comparative analysis down the road. Going into a sample, I'll pick a shotgun sequencing sample to start with. Pick this one and go in and view the bacterial results first. So this is a sample that we've uploaded and analyzed already. So if we go in and we can click the eye to see the metadata, this one's been filled out already. So it's a mixed DNA sample of a mock community. The sample source is Baylor College of Medicine. The sample's from May 10th, 2010. It's part of the Human Microbiome Project. It was a lab-made sample. So you can understand the power of the metadata and being able to go back to and reference the metadata for your individual samples, and then in the future be able to sort and select those samples based on metadata. So just an overview of the interface. In this sample, there's 48 organisms found, about 8 million reads, and this is based on the bacterial results, and these are unfiltered. Dr. Colwell spoke a lot about machine learning earlier, and we've done a lot of machine learning to improve our results, reducing false positives and reducing false negatives. So when you go into the filtered results, that's where the machine learning kicks in, and it reduces the number of organisms down to 28. And these organisms are the ones that we're very confident are in the sample through multiple statistics that the algorithm takes into account in the back end to create cutoffs of confidence. So once you have your sample and you analyze it, you have a table view. You're able to see the different organisms. Everything's sortable. You can sort by frequency, unique match percentage, total match percentage, relative abundance. Relative abundance is the relative abundance of that organism in the sample. So all of those relative abundances sum up to 100%. Unique match percentage and total match percentage. Total match percentage is a surrogate of genome coverage. So it's the amount of coverage of that specific genome. Unique match percentage is of our database, the unique biomarkers for that specific strain or that organism, how much of those we found. And then the frequency is the number of hits that were found for that organism. You can also click on the tax ID, and that'll take you to NCBI, so you can dive in deeper on those on that individual organism. So that's the table view. You can export this table view and open it up in Excel, play around with it as you wish. We also have a lot of beautiful visualizations that are built in. So this first one is a sunburst chart, and you can deep dive different taxonomic levels of your sample. It's interactive. Everything's exportable, so you can export to PNG and SVG, which are vector graphics. And it allows you to really explore your sample in depth. We have a bubble chart, and this bubble chart is based on relative abundance. So the bigger the bubble, the more abundant that organism is in your sample. We have two different tree views. This one's the collapsible tree view. It's interactive in the sense that you can open and close nodes and customize the view as you like to see it. And then, of course, you can export it and use it in a presentation or in a publication. We also have a radial tree, and the radial tree is a single view of your tree from your sample. So it's a much easier one snapshot view of all the organisms in your sample. Two new visualizations that we've added are a donut chart. And this donut chart you can look at based on frequency or relative abundance, and you can switch them. You have the ability to adjust the tail. So it defaults to 20 organisms. In this case, this sample has less than 20, but if you wanted to show all of the organisms, it'll add those in and remove the others, which is the tail. You also have a stacked bar graph. So you can view this as a stacked bar graph and similar functionality to be able to switch between frequency and relative abundance, and also the ability to adjust the tail if you want to. So if you say, I want the tail to be 0, all of the organisms show up or don't show up. And so that's the bacterial results. Now, since we also have fungal results, so you can switch the database and look at the fungal results. And similarly, you'll have a table view and you'll have the different visualizations that we just showed for bacteria. We have a protist database. We have a virus database. And we also have antibiotic resistance genes. So if you go into antibiotic resistance genes, you'll see a list of the genes that are found in the sample, and your total match percentage would be your genome coverage for those genes. And again, these are able to be filtered and unfiltered. So the filtered ones are the ones that we're confident about, and the unfiltered ones you would need to validate with some additional orthogonal validation. And finally, we have virulence factors. So virulence factors work similarly in that we have the listing of the genes and then also the gene coverage for the virulence factors. And virulence factors are genes that are associated to how pathogenic an organism is. So moving on, so that's one sample, and one sample is great to look at, but when you're doing a study, one sample really isn't representative of how to do discovery and how to come up with findings. So we also have the function for comparative analysis, and I'll quickly put one together to show you how fast it is and how it works. So if you hit create, we would name it, and we pick a database. So in this case, I'll pick bacteria, and I'll look at it on the filtered results based on frequency. And since we have about 60 human microbiome projects loaded on every account, so everyone has access to them and can play with them, I'm going to choose some of those for a quick demo. So I'll choose some stool samples, I'll choose some oral samples, and I'll choose some vaginal samples. And you can search through your samples. So in this case, I'm going to search my stool samples and pick some of the stool samples. I'm going to pick some samples and put them in the oral cohort. So these are tongue, and let's say we have some plaque samples. I'll put those in oral as well. And some vaginal samples. So, and all you do is you hit submit, it takes about a minute for the comparative analysis to be put together, and what it does is it builds a large matrix of all organisms found in all samples against all samples. And then once that analysis is complete, we have different visualizations that I'll be able to show you, which include some of the output that Dr. Colwell just showed us. So it's just opening up now, and here again you have this matrix table. You can export the table if you wanted to analyze it in another package. If it's Excel or if it's R, that can be done. We have a heat map view, and so you have the ability to look at it from a heat map standpoint. You can switch between taxonomic levels. So if you want to look at it from a phylum standpoint, you can look at it at a phylum standpoint. You can switch to order, down to strain. And then we also have a stacked bar graph, which you can switch between absolute and relative. You can also switch between log scale and no log scale. And you can randomize the colors if the gradient is too similar between them. And again, you can switch between taxonomic levels here as well. So this gives you the ability to really compare samples side by side and see how similar or how different they are. My favorite one is the 3D PCA. So we have a 3D PCA, and this really gives you the ability to quickly see what samples are similar, what cohorts exist, and what samples are deviant. So in this case, these are the stool samples. They're pretty similar. The vaginal samples cluster really tightly, and the oral samples looks like there's kind of two groups here. In addition to those, we have alpha diversity, and you can look at it in three different metrics. One is Chow-1. You can look at it from the Simpson and the Shannon side. We also have beta diversity, and that can be done through Jacquard and Bray-Curtis. And finally, some just general QC information in terms of read statistics, so you can understand how similar your samples are in terms of the read depths that they were put through. One last example is just in terms of scalability. The platform is very scalable. So we've taken about 400 healthy microbiome samples from around the world and then run them through. So I'm switching over to an analysis we've already run, and this might take 2 or 3 minutes longer to put together because it's about 400 samples, but this is a USA healthy microbiome cohort. And then you can look at China, and that's what China would look like, and you can add that in. Japan clusters pretty well together there. Native American is pretty spread out, but you'd imagine that different areas, Native Americans come from different parts of the country, so it might make sense there. Tanzania, adding in Spain, Denmark, and even rural India. So very powerful. You're able to compare many samples at the same time and really get a feel for how similar or how different they are and what samples you might want to dive into deeper. So for example, this Native American sample is very different than the rest of the entire world. So this would be an interesting one to look at. Layering on top of that, if you go in and we add in a disease cohort to that same healthy cohort, the healthy cohort looks very similar, and adding in a Crohn's disease cohort on top actually shows how different and how variable the Crohn's disease cohort is to any sample in the global healthy microbiome, whether from USA or India or Tanzania or Denmark. And I think this really gives you the ability and the power to quickly dive into microbiome data, quickly analyze it, and be able to action the data rather than spending a lot of time generating the data. And that's really where the power of our platform comes in. So thank you for listening in on the demo, and thanks for listening in the webinar. And I think we're ready for some questions.
Thank you, Manoj. Thank you, Dr. Colwell, as well. I think this is a great presentation. Again, there are resources available on the right margin.
Just to show you the speed. So the one that we uploaded earlier was a 20 NIDX, and it's already analyzed. I forgot to come back to it. So that was it. I'm done. Thanks, Kelly.
Great. No, I appreciate it. I think it gives everybody the power of understanding of how quickly things, even with a slow internet in a hotel room, you can upload and analyze data. So this is great. So with that, I'll kick off a few questions. Just a reminder, you can ask questions in the Q&A widget on the screen by typing in your question in the field there. So Manoj, let's start with you. How important is strain resolution, and why do you perform, or how do you perform better than others in that specific aspect?
I think I'm gonna defer this one to Dr. Colwell, since this has been the basis of everything she's done.
Oh, yay!
Right. All right. That's a good question. It's important because when you're making a diagnostic call for public health application, you need to know, for example, if you've got E. coli or E. coli 0157. Very different outcome for the patient. For example, specific strains of strep mutans produce hemorrhagic damage in the brain of mice and other tissues, and other strains are risk factors for ulcerative colitis. And another example I could give is the strains of Toxoplasma gondii have different pathologies and cause different host responses. Variants of Staph epidermidis and Staph aureus have effects on virulence and whether they can form biofilms. I could go on and on. The strains of Bifidobacterium longum, some protect against pathogens like E. coli, and others have immunomodulatory properties. So I think, just to answer the question directly, these examples point out why subspecies and strain level identification is really, really critical to understanding microbial symbiosis, dysbiosis. It demonstrates the power of our metagenomics because we get really fine taxonomic resolution. This is critical information in microbiome research for epidemiology and forensics, outbreaks, and so forth. So I think the reason why we've got better performance in our system is probably due to the fact that our database is phylogenetically organized. So we have distinct clades that represent the distinct subspecies, like, for example, pathovar, cerovar. So our database not only is highly curated, but it provides really this kind of precise subspecies identification.
So that's a great segue into the next question, which is, what's the advantage of organizing the database as a phylogenetic tree as opposed to a taxonomic-based organization?
Well, there are a lot of advantages, but here's a couple. It really helps in the curation process. If the taxonomy of a genome is misrepresented, like if you sequenced E. coli, but you had it labeled as Klebsiella, and you submitted it to a public database, with our phylogenetic analysis, we detect the inconsistency. We correct it. Secondly, it's an opportunity to achieve subspecies and strain level identification. In any taxonomic scheme, you get species and then strains, but most of the critical information, particularly for pathogens, is in the subspecies, like the cerovar, the pathovar, the biovar. And the phylogenetic tree gives specific clades for each of these, and it allows this classification and identification at a much, much finer level. And then thirdly, I could comment that when you get a novel strain or species that's maybe not in the database, this approach, phylogenetic approach, helps identify the phylogenetic near neighbor. And that's very important because that allows you to hone in on it. And then I guess one more comment I could make finally. We use the genomic data as a query and we probe those data against the genomic genomic databases. So we're not using any phenotypic data, and so it makes a lot of sense to organize the database in a phylogenetic tree. I hope that answers the question. Thank you.
It does, and we have actually quite a few questions that are queued up. So, but we are a couple of minutes over at this point, so we're going to have to follow up with each one of you directly. Again, thank you, Manoj Dadlani and Dr. Rita Colwell, for your presentation today on CosmosID. Thank you, everyone.
Thank you.
Thank you.